Hello everybody. In lecture 6 we will cover the topics of proteins as molecules and proteomics as a science. The learning objectives of this lecture are to examine the variability of proteins, their connection to human diseases, how protein structures define their functions, and make the audience familiar with the definition of intrinsically disordered proteins. We will start with an overview of proteins and proteomics. We'll talk about protein synthesis process and structures, role of proteins in human diseases, specifically how the intrinsically disordered proteins cause diseases, methods of proteomics, and where the information about proteomics can be mined from. As a group of molecules, proteins were described in 1838 by a Dutch chemist Mulder and by a Swedish chemist Berzelius. Berzelius coined the term as proteins, and he also authored the elemental composition of proteins empirical formula. The name of proteins come from the Greek language, meaning primary or principal. And by the way, one of the proteomics analysis and data management tools is called Proteus. Regarding the sources, proteins are omnipresent, being part of almost all food that we digest except for sugars, oil, water, and salt. Although different food types supply a variety of proteins and amino acids, legumes and fruits are scarce in several essential amino acids, and not to put the rain on the veggies parade. For example, threonine and tryptophan content is very low in cereals and methionine in legumes. Some other sources of proteins and amino acids include microorganisms and spores. In cells, proteins make up to 50% of dry matter, thus it is an abundant source for proteomics research when proteins are isolated from cells. Proteins have a multitude of direct or indirect functions in cells, and it could be said that basically all functions of a living organism are mediated by proteins. Among protein functions, gene expression is probably the most basic because proteins participate in all steps of cell multiplications. Proteins also regulate the metabolic events via enzymes and hormones, serve as transporters in fluid circulation and between cells compartments. From a clinical point of view, proteins are the basis of the clotting cascade and participate in immunological responses to infections. Proteins are structural units of the contractile tissues such as muscles, and neurotransmitters are basically proteins with specific functions. The connective tissue is also composed of proteins, and finally proteins serve as reservoirs of amino acids and nutrients. The list is far from being all-inclusive, and each of the described functions probably could be extended under specific conditions and environment. The largest protein is titin, also called connectin, which is also the third most abundant protein in eukaryotes after myosin and actin. 8 isoforms of titin have been annotated at least partially, the largest consisting of 363 exons and having 17,106 base pair in the longest exon. The existing isoforms range from 25 to 35,000 of amino acids in structure, and have molecular weights of more than 30 million daltons or several thousand of kilodaltons. Molecular weight of proteins is a very important characteristic, and it is calculated based on specific amino acid composition. There are many websites with built-in molecular weight calculators that account for amino acids and nucleotide sequence compositions. However, a more simple and practical formula to calculate the molecular weight of a protein is to multiply the total number of amino acids by 111. In biomedical research, knowing the molecular weight of proteins may help you estimating the rate of migration of proteins on the gel, for example. Based on the number of amino acids and coding principles, in theory, there may be a huge number of proteins. It is estimated that there are quadrillions of proteins in nature, and that there are about 1 million different proteins in humans, albeit not necessarily the same proteins within a single cell. Dr. Ron Miller of the Weizmann Institute of Science estimated in 2013 that each cubic micron of a cell may have several million molecules of proteins. Well, any estimates are probably very speculative, considering the proteins are dynamic structures and they may have many forms and shapes and it will take probably very long time to bring the proteomics to the level of understanding that we have in genomics. Published in 2014, the human proteome map described only around 20,000 or a little bit over 80% of human genes coding for proteins. Surprisingly, aside from 80–90% of proteins being coded for by genes, as we define them, another 10% of proteins are being coded for by the long intergenic non-coding RNA or link RNAs. For the purpose of our class, we still consider that proteins are coded for by genes, but we need to keep in mind that some of them may be encoded for by the non-coding RNA, no pun intended. In general terms, proteomics is a branch of systems biology that studies the protein's properties, structures, functions, modifications, interactions with other proteins or biological molecules as a global view of cellular processes at the protein level. Proteomics term was coined by Mark Wilkins in 1994 as a combination of words protein and genome to encompass all proteins that could be produced through the genome from translation, alternative splicing, and post-translational modifications. The term of system biology defines the studies of interactions among all components of biological molecules as a dynamic process. Proteomics plays a fundamental role in each of the major components of the system biology, as proteins influence and are being influenced by every other type of molecules. A better understanding of proteomics is necessary for a progress in the system biology. As we know, proteins consist of amino acids and are chemically classified based on their R group charges at a neutral pH into charged positively and negatively, and neutral, polar, or nonpolar. Proteins are synthesized within the endoplasmic reticulum, or ER, of cells by translation of messenger RNA on the large subunits of ribosomes, which attract the respective transport RNA or tRNA with specific amino acids attached. While we know that proteins are coded for by genes, we need to keep in mind that there are exceptions, such as link RNAs, less studied regions of sequences that are conventionally called non-coding and may have a number of other functions less understood at this time. The sequence of messenger RNA codons establishes the order of amino acids assembled by the ribosomes. tRNAs carry the amino acids one triplet per one tRNA molecule at the amino acid attachment site, and the anti-codon loop of tRNA is capable of recognizing divergent codon bases, and we need to be aware of the wobble base pair effect. The wobble base pair effect was proposed by Watson in 1966 to explain the multitude of amino acids recognized by a single tRNA. First two bases of codons are specific for each triplet of the anticodon. The third base of the anticodon is the source of the wobble effect, as it can be substituted by a non-conventional pairing or wobbling. If the anticodon starts with A or C, counting from 5 to 3' direction, the pairing is specific, and if the starting nucleotides are U or G, the pairing is less specific due to the wobble effect. One of the major nucleosides involved in the wobble effect is hypoxanthine, a nucleobase of inosine or ribofuranose. Inosine is synthesized in the process of adenosine transformation into inosine monophosphate, which is then catalyzed into inosine. In addition to the possibility of both U and T pairing with G, inosine may pair with A, C, and U, providing a diversity of codons that could be carried by the tRNAs. Knowledge of inosine metabolism has led to advances in immunotherapy in recent decades. Inosine monophosphate is oxidized by the inosine monophosphate dehydrogenase enzyme, yielding the xanthosine monophosphate, a key precursor in purine metabolism. The mycophenolic acid, or mycophenolate mofetyl, branded as Cellcept by Roche or Myfortic by Novartis, is an anti-metabolite, anti-proliferative drug that acts as an inhibitor of inosine monophosphate dehydrogenase. It is used in the treatment of a variety of autoimmune diseases caused by leukocytes hyperproliferation. The uptake of purines by actively dividing B cells can exceed 8 times or more that of normal body cells and cannot operate the purine salvage pathway. Thus, by inhibiting the purine synthesis, the mycophenolic acid slows down the invasion of leukocytes in immunological disorders. Mycophenolic acid is also used as immunosuppressant for preventing acute rejections of organ transplants. However, its prolonged use may increase the risk of lymphoma and other malignant tumors, and the drug is teratogenic in animals. So again, the chain of events in purine synthesis is interrupted by the mycophenolic acid inhibiting the inosine monophosphate dehydrogenase. The methods of proteomics are quite diverse and are discussed in the textbook. Mass spectroscopy is one of the major methods of analysis of all peptides composition in solution, and identification of peptides is performed by calculating the mass to pi ratio. In short, the mass spectrometry methods start with digestion of a mixture of proteins, followed by their separation by size, fragmentation, and analysis or identification of peptides. As you can see, the two approaches as top-down or shotgun have many similar steps and end up with the same process of identification. If the separation of peptides by sizes is less evident, the identification of proteins may be less precise, particularly in certain post-translational modifications where the mass difference is in single digits. Usually, the pi versus size ratio is imputed into databases with defined or calculated numbers corresponding to each peptide and their post-translational modifications, and the peptides are identified as the most likely to occur in cases the numbers are close between more than one peptide. One of the fastest methods of quantification of total proteins is based on the nitrogen content in examined samples. In proteins, nitrogen makes up to 16% of all elements. A sample is heated for several hours at 300 degrees Celsius, treated with concentrated sulfuric acid, resulting in a conversion of nitrogen into ammonium sulfate. Further treatment of the ammonium sulfate with a strong base, such as sodium hydroxide, will release ammonium, which is ultimately titrated, and the obtained number is multiplied by a coefficient to obtain the total protein content in percentage units. The method was invented over 100 years ago, and it is still used in many countries around the world. The main flaw is the assumption that all nitrogen molecules come from proteins in a sample. That is what happened in the infamous Chinese milk scandal in 2008. As you may remember, over 300,000 victims were affected, with over 50,000 hospitalizations due to kidney stones and failure. China is the major consumer of the powder milk as part of the baby food composition. When many infants developed kidney stones, the melamine was identified in the milk powder upon a detailed examination of samples. Melamine is a type of a plastic resin with flame retardant properties, which has a low toxicity to living organisms, and it has been used as a source of nitrogen in foods for domestic animals. Inside living organisms, melamine reacts with cyanuric acid, both non-toxic chemicals, resulting in formation of insoluble crystals in kidney, forming stones and leading potentially to kidney failure. So the total protein content in samples was masked by the nitrogen molecules derived from melamine instead of pure proteins. We are familiar with structural conformation of proteins such as primary, secondary, tertiary, and quaternary. Proteins tend to take a conformation that requires the least amount of energy to maintain, and such a conformation is called a native state. The number of potential conformations of proteins are enormous based on the possible combinations of amino acid triplets. The most widely used conformations for protein analysis are their primary structures and 3D crystallization forms. The reason are obviously in our ability to fix the forms and analyze them. The protein structure is constantly changing from one form to another, and the 3D structure can be fixed as crystals and visualized by X-ray or NMR methods. We need to keep in mind that even small modifications in primary structures, that is, in amino acid sequences, may result in large functional changes. There are many examples when a single mutation in an amino acid or in the coding triplet is the cause of a disease. One of the most recognizable diseases caused by a single mutation is sickle cell anemia, in which the valine in position 6 of the hemoglobin beta chain is replaced by glutamic acid. Basically, the primary structure can be determined empirically or experimentally by sequencing of the amino acids using proteomics methods, or, as in the majority of proteins, it could be theoretically deducted based on their functional domains and motifs. As we know, proteins are defined by their 3D conformational structure. The intrinsically disordered proteins, or IDP, however, have no defined CD structure in solutions, as they represent ensembles of their secondary and tertiary conformations in regions of at least 30 to 50 amino acids long. The term IDP has a number of synonyms, such as rheomorphic, natively disordered or unfolded, unstructured, all of them representing the lack of stable conformation in solution. Whether a protein is structured or not could be deducted from protein amino acids composition, the IDP being enriched in polar disordered proteins types of amino acids in addition to the structure-breaking hydrophobic proline, and IDP are depleted at the same time of order-promoting residues. This slide represents a proposed sequence to structure to function that could explain the novel functions of disordered proteins. As you can see, some of the functions are not attributed exclusively to ordered or folding proteins, but are most likely shared among different groups in dependence on specific conditions. The classical function of lock and key is more typical for enzymes, based on which the principle was described when the proteins were discovered back in the 18th century. As with everything in nature, the intrinsically disordered proteins have some advantages and disadvantages compared to the structured in their native state proteins. For example, IDPs have larger interaction surface area compared to globular proteins of similar length. Their conformational flexibility and exposure of active domains allowed for scaffolding and interaction with a large number of proteins, and a variety of post-translational modifications altered their functions and stability in cells. Also, many IDP fold upon binding, and their interaction is relatively highly specific and has a low affinity, meaning that they can bind precisely to their targets and dissociate rapidly as the signal has been delivered. Translation of ordered proteins ends up in assembled and folded structures exported from the ER into cytoplasm. The ordered proteins are folded into their native states and exported into cytoplasm. However, the disordered proteins in many cases are being degraded in ER, and when exported are being post-translational modified by a number of binding factors and proteins. Examples of the numerous classes of IDP include MYC, P53, and axin, which are some of the major players in many human diseases, including cancer, autoimmune disorders, and are involved in more than one pathway depending on their localization and specific post-translational modifications. The textbook has some outdated information on the protein databases. For example, the International Protein Index database is no longer supported, and some links have changed. This slide provides the current list as of June 2015. Again, some of the databases contain raw sequences submitted by researchers, and a few of them have the data curated or experimentally validated. The information presented in databases differ independent of the scope of collection, but usually the protein databases have their 3D structures, interaction drugs, chemicals, and other components in addition to just sequences of amino acids. Many databases have tutorials on using them, as it may be complicated to find and apply all features provided by the developers and scientists. The link on this slide will take you to the short tutorials on using the Protein Data Bank portal. So this concludes the Lecture 6 presentation, and if you have any questions, please post them in the discussion board or send me an email. The additional files provided for Lecture 6 is giving you more information about the topics that may be covered in the exam.